أهلا بيكم في دروس الأحياء للصف الثالث ثانوي لغات. Today we will continue answering the questions about the DNA and nucleic acid curriculum. So let's begin. Compare between DNA and RNA nucleotides. A nucleotide of RNA consists of ribonucleotides due to contains ribose sugar, so it's called ribonucleic acid. And usually it's a single strand, but it can form double sections. The nitrogen bases are adenine, guanine, cytosine, and uracil instead of thymine. Uracil base pairs with adenine. On the other hand, a nucleotide of DNA consists of deoxyribonucleotides due to it contains deoxyribose sugar, so it's called deoxyribonucleic acid. It's double strands, two complementary strands. The nitrogen bases are adenine, guanine, cytosine, and thymine. Thymine base pairs with adenine. Okay, DNA in prokaryotes and eukaryotes. DNA in prokaryotes, like the Escherichia coli, for example, this is a bacteria in the human beings, is one double helix of DNA, circular DNA, not complexed with proteins, and is attached to plasma membrane at one or 2 points, and is folded many times. If DNA of Escherichia coli is stretched out, it will be 1.4 millimeter long, whereas the cell is about 2 micrometer. DNA in eukaryotes is like a human somatic cell, for example. The chromatic material divides into chromosomes. Each one has a single DNA, linear DNA in shape, complexed with proteins, not attached to plasma membrane where it's found inside the nucleus, and is folded many times. If the DNA of the human cell is stretched out in a straight line, they would be about 2 meters, although the size of the nucleus is about 2 to 3 micrometers long. Okay, this is the comparison between DNA of prokaryotes and eukaryotes. Structural and regulatory proteins. Structural proteins constitute the building materials in living organisms, such as the actin and myosin found in muscles and other contractile systems, such as collagens that form connective ligaments, such as keratin that protects the skin, protective covering of the hair, skin, claws, and horns. Regulatory proteins that regulate the various processes and activities in the organism, such as enzymes that regulate chemical reactions, antibodies that provide the immunity against infection, hormones that help, like the nervous system, it helps organism to respond to the constantly changing changes in the internal and external environment. Replicated and recombinant DNA. A replicated DNA needs temperature and forms from 2 complete DNA or RNA strands, and need different sources of DNA and RNA, and used to determine the evolutionary relationship between different species and detect the presence of a particular gene in the genome. While recombinant DNA needs to restriction and ligase enzymes, forms from 2 different segments of DNA, needs 2 different sources of for DNA, and used in the molecular technology in the medical field, agricultural, and industry. Question number 11. Most of the studies concerning the discovery of the true genetic material used viruses and bacteria. Explain one of these experiments where the viruses and bacteria were used to prove that genetic material is DNA and not protein. Remember the experiment we talked about injecting a bacteria with a bacteriophage or plasmid and using radioactive sulfur and phosphorus. Remember the nucleic acids contain phosphorus while proteins contain sulfur. So you provide radioactive sulfur and phosphorus to determine which is responsible for the transmit or the passage of genetic material. Is it proteins or phosphorus? This is what Hershey's and Chase's experiments did. They labeled phage DNA with radioactive phosphorus and phage protein with radioactive sulfur and allowed the radioactive phage to infect the bacterium, and they determined the amount of radioactive phosphorus and sulfur inside and outside the bacterium. They discovered that the radioactive phosphorus always entered the bacterium, whereas less than 3% of the phage protein with sulfur radioactive sulfur entered the bacterium. This was strong evidence that DNA is the genetic material of phage and not protein. Okay, 12. What is the importance of the human genome? We studied this. I told you that identifying the genes cause for rare common hereditary diseases, identifying the genes causing disability of organs to perform vital activities, preparing drugs without side effects in the future, and studying the evolution of living organisms by comparing the human genome with genes of living organisms, and improving the offsprings by identifying the defected genes of the fetus before it's born and how to be cured. It's called gene therapy. This is the importance of the human genome. Last question is briefly explain the steps of protein synthesis, starting with the transcription and genetic information. Remember the two subunits of ribosome, when they come together in the presence of mRNA, adjusts itself to the P site, peptidyle site. Remember that? The RNA polymerase enzyme binds to the sequence of nucleotides on DNA called the promoter, where the two strands of DNA are separated. Then RNA polymerase move along the DNA and joins the complementary nucleotides اللي هم يعني الأدينين والcytosine والguanine والuracil, one by one, starting from 5 to 3 direction on the growing strand, forming mRNA that moves to the cytoplasm. Now the mRNA binds to a small ribosomal subunit and first codon AUG. This codes for the formation of methionine that pairs with anticodon of tRNA carrying methionine, and that's why methionine is the first amino acid in the polypeptide chain. Then a large ribosomal subunit binds to the complex compound where the second tRNA binds in, remember, the A site of the large ribosomal subunit, and the tRNA anticodon complementary to the second mRNA codon. And now an enzyme called peptidyl transferase reaction binds the formed amino acids with peptidyl bonds between them to form the polypeptide chain. And the large ribosomal subunit moves along mRNA, so the second tRNA carrying the peptide bond moves from A to P site, and the A site will become empty to receive the third tRNA with the third amino acid, and so forth. When reaching a stop codon at the A site reaches a stop codon releasing factor binds to the stop codon and causes the mRNA to leave the ribosome, and the ribosomal subunits separate where the polypeptide is terminated. But I told you before that once mRNA emerges, it becomes attached to many ribosomes, transcripting its message, and this is called polysomes or polyribosomes. This concludes the answers of the questions about the curriculum of DNA and nucleic acids. أتمنى لكم التوفيق, and this concludes the curriculum of biology for the third secondary international schools. I hope you got the maximum benefit from the course and from this lecture, and thank you very much for watching.